Introduction to protein function. Again, these slides you have seen. Thanks to Janai, some of these introductory slides come from Janai Ofran, who is now at Biolan University, Marco Punta, who is now in Paris, Avner Schlesinger, who is, oops, that is not new, who is now in Mount Sinai, Kaz Wrzynski, who is also not true, is now in the Genome Center in New York. Back to New York. The basics of life, the idea is that the common element to life is DNA, and that in fact today we believe, where Leonardo da Vinci believed in shape is the answer to understanding how life fits into the universe, now we believe is DNA. How did life begin? What's the molecule that started life? Anybody has heard of? Yeah?
No, I remember that Miller experiment.
And so we get into this point here. So the assumption, it got us Nobel Prize, and this is just the book where I took it from, but the Nobel Prize, Tom Cech got his Nobel Prize for something else. The Nobel Prize ultimately was the Ig Nobel Prize in the 60s, with the idea that life starts with RNA. Essentially, they started it early on. They tried to show it already in the 50s. We are not, since the 60s, I don't believe we have gone very far. There's a lot of science going on here. We still have not had really the breakthrough, but it is now, I believe, the accepted model. Life starts with RNA. Again, 40 years later, we still don't have a proof, but we believe more in this model. In this model, RNA, in fact, the molecule that is also able to self-replicate and have some activity, begins to sort of use DNA and then forms proteins. Actually, the idea of forming proteins is that there are certain processes, RNA enzymatic activity, that RNA cannot do alone. It brings in peptides, amino acids, and that in fact today's selection of 20 amino acids is because the RNA needs to enslave amino acids or peptides made up of amino acids that do the enzymatic activity that it cannot perform on its own. Okay? If so, then you would expect that in today's proteins, you somehow see some signs of this old RNA binding, or this RNA helping activity. And that's exactly the work of Andrei Lupas, who goes into the universe of fragments of folds today, and tries to verify the traces of the ancient RNA binding slave life in some sense. He begins looking at sequences and structures, begins to simply take it out from looking at similarities, and then does experiments, and has dedicated the last 20-something years to advance this concept more and more and more. And I believe for several domains, and one of those sets of domains are shown here, his story is very, very convincing, has become very convincing. So that again, what we see today in today's protein structure is a relic of the old universe of RNA. Okay? But then today we have the information flow from DNA to RNA to protein. The dent in the picture, of course, is that RNA is not an intermediate. As you all know, there are long non-coding RNAs. So they're short RNAs that are for regulation, very, very important for regulation. What is short? When I talk about short RNAs, any idea what these typically, the regulatory RNAs, siRI, they come in very, very, very many different names. But any idea what typically short is? 200? No, short RNAs are much shorter than that. So they're in the ballpark of 20. So 300 is already, 300 nucleotides is what people would call long non-coding RNAs. And 300 is a good number for that. Sometimes, so there's a little shifting here. Sometimes when you have 100 nucleotides, people already talk about long non-coding. But 300 is clearly long for non-coding. Does anybody know whether we have anything like that? Do they exist? Ribosome, for instance, is full of proteins and RNAs that are long non-coding and that are essential for the function of the ribosome, is one example. But if I asked you, how many do you believe we find? So I'm going to show you a number from an experiment that was published 10 years ago. And in that experiment they did mouse. It's from Riken. Mouse has roughly 20,000 proteins. How many long non-coding RNAs do they have? And I believe the threshold there was 100. Any idea? 100? 10? 1,000? The surprising answer is hidden. Another one. So the figure is nice but unneeded. Oh, it crashed. No, it didn't. Here we are. Single paper in Science in 2005 with 140 something authors. They had a total transcript of 180,000. New protein coding, 16,000 in that. New proteins, 5,000. Multiple splice variants, 65%. Is actually a lower number. But the most important number is not on here. The non-coding part here is about 20,000. So long non-coding in mouse, essentially, before that paper was published, people believed there were tens, maybe 100. And this paper said there are 20,000, which was, in fact, in the ballpark of the proteome. And they are not identical. So how come we need so many? We still don't know. That was clearly a big surprise. RNA is not an intermediate, but for the remainder of the lecture, I will nevertheless treat it as if. So typically the idea is that proteins interact because they have particular shapes, structure. That was the first part of the lecture. How does sequence determine structure? Now the second part is really how does structure or how does sequence determine function? What is a protein? What is a gene? A gene is a consecutive structure on a genome. What is a protein? Now let's begin with the following. We cannot say a protein is the thing that is expressed by a genome. Already in this experiment before, you saw this alternative splicing. Okay. Can we say that whatever comes out of one genomic region is a set of proteins? Or one moment, let me turn that around. If I have a protein sequence, is this protein sequence always mapping to the same gene? Is the protein coming only from one gene? No. There are proteins that are spliced together from two genes? Because this is biology and everything is possible, or what? Fair enough, you're right. There are, in fact, many examples where you are spliced together from different chromosomes. Not only different genes, so the genomic material can be spliced together from very, very different regions. That makes this question such a complicated one. Because now you have the issue that when do you call something one protein as opposed to a set of proteins that collaborate or that interact, right? When is it a unit that is one protein? And when is it a set of things? And co-expression typically is in fact the answer. Now, let's get back to, in fact, it's not that simple, is what I'm saying. But ultimately, we come to a point where we have a protein sequence. The protein sequence translates to a protein structure. Here are some examples. Here are more examples, in particular channels, membrane proteins. And typically, bioinformatics or computational biology or experimental biology always starts to look up similarities to other sequences, and by these similarities, in this case of cytochrome P450, if I did not have the annotation here of cytochrome P450, and I would find this in the database, then I would assume that this, because of its sequence similarity, is also cytochrome P450, and that in fact it acts the same way, with tiny differences. And ultimately, these tiny differences, that is the protein that is the taste, that makes the taste. So spearmint and peppermint taste slightly differently. So these sequences do the same thing, but there's something they induce in our tongue that is specific to either or. Okay? But this is the subtle difference. Essentially, they're both cytochrome P450s. Both of those essentially create the same sensation, do the same function. Now that brings us to the question of protein function. Can you define that? How would you define protein function? What have you encountered that are attempts to define protein function? Maybe I asked my question the wrong way somehow. The quiet majority of 12 people should have determined that they want to sleep, because that's what they have. It's not true. I'm sorry. I'm trying to push you. You have never heard any classifications of protein function? Guys, you must have heard some. No? Yeah. Yes. And I'll get to that in a minute. That is the most dominant one. Here's one I begin with that is slightly simpler. So alcohol dehydrogenase, ADH5. This particular version here is from human. By the way, there's one aspect of its function that is very crucial. Those are two it's a homodimer. What it means is you have two versions of the same protein duplicated. When you have the term homodimer, typically it means it's a homodimer for functional reasons. That means they have to come together. Again, these are the same proteins, two versions of the same protein. If you expressed only one, you would not have the function. You need to have some abundance expressed, they need to work together, then you get the reaction. The reaction is an enzymatic reaction that, as this text here shows in more detail, the first part of the text is an EC number. Anybody heard that term? That's the enzyme classification. This is not the right translation for the C, but it makes it simpler to understand what it is. And this is essentially a number. The number classifies the first digit, and we will get in more detail back to that. It's like a hierarchy. And this hierarchy has essentially to do what you do is there are certain types of substrates enzymes bind, certain types of reaction, and you simply classify all substrates or reactions, and you give out a hierarchy of numbers. Then you can, okay, so far, so good. Complete hierarchy, every enzyme has one number, okay? That's the number. Now, further down, you see, yes, that is the enzymatic reaction. It catalyzes the oxidation of primary and secondary alcohols to their corresponding aldehydes through the mechanism that involves removal of hydrogen. Oh no, it's not here. It's on the next slide. I'm sorry. Now, this again is in more detail. Here's the chemical reaction. But this is part of the pathway. So if you don't have that thing, then it's very difficult for you. If you don't have ADH5, it's very difficult for you to digest alcohol. This is why when people who lack this, and there are Asians typically who don't have ADH, when you lack that, it's much more difficult for you to digest alcohol. But the breakdown of alcohol is a longer path. It's a longer set, and you already said that, so association, we typically have processes over more than 7 proteins, and this is KEGG, the Kyoto Encyclopedia for Gene, whatever. 25 years by now, I believe, of simply taking all the known material about every single process that is relevant for life and put it into graphs like the one on the right-hand side. 25 years, 100 people per year, got us KEGG. It's the most outstanding resource for pathways and reaction mechanisms, and every single point in here is a reaction. And the ADH5 is just one little point here. But being a point in this pathway is another aspect of its function. Okay? It's not only that it's a particular enzyme that has a particular enzymatic activity and it does catalyze a particular reaction. It's also that it's part of this pathway. Now, in fact, you could argue protein function It seems very intuitive, but it's not well defined. You could argue the function of a protein depends on how it binds the atom. You could argue that essentially the type of enzymatic activity, the way it does its enzymatic activity is important. You could argue the cellular component, so whether it's a kinase or not, the developmental, when is it regulated? How is it regulated? Physiological is related to disease. Genetic, it's dominant or recessive. Which of those you consider function depends on your interests. Geneticists have the interest here. There is no answer to what is protein function. It's all of that. For you, it's relevant where you live. This lecture will focus here on the biochemical cellular part. And for us, typically, whatever happens to or through a protein is function. And I believe we cannot wrap this up in a minute. I believe that's the end of the lecture, and I will continue here when we get into sequence structure and motion. Thanks for your attention.